Assalamu alaikum everyone. Welcome back to my channel, Amara's Insight. In this lecture, we are going to talk about the ubiquitination. What is ubiquitination? What is ubiquitin? All these things will be discussed in this short lecture. So, what is ubiquitination? Question is, ubiquitination is basically a post-translational process. It's a post-translational modificational process where the protein specific protein ubiquitin is attached to a target protein. So if we, let's see, we have this protein. This protein is basically a complete protein that has been formed after the process of transcription, after the complete process of the translation. Now, if we want to modify this protein by the ubiquitination tag, then this process is basically the ubiquitination. For example, what does that mean is if this complete protein that is formed after the translation, this protein is you can say it's a kind of a damaged protein. It's a misfolded protein. It's kind of a unfolded. Now this protein is not useful. This protein will not good for our cell. It will be a dangerous for our cell. Now this protein needs to be degraded. So how this protein will degrade? Now we need to modify this protein. What will we do? We'll add ubiquitination tag. We'll add ubiquitin protein to this particular target protein. Now this protein will be degraded after the ubiquitination tag. This is the ubiquitination. Now, ubiquitination basically involves the covalent attachment of the ubiquitin, which itself is a small protein, to a target protein. Now, what is the target protein? Now, target protein is that protein to which we add the ubiquitination tag. The target protein is the one with that needs to be degraded. That is actually dangerous for cell. That is actually a damaged protein. That is actually misfolded protein. This protein that we want to degrade, that is not useful for the cell, that we want to modify by the addition of the ubiquitin. This particular protein is referred to as a target protein. Now this process of the ubiquitination, it plays very important role in regulating in the regulation of the various cellular processes. This basically includes the first of all, it is the protein degradation. The protein that is damaged, that is need to be degraded. So this process is very important for that. And next is the it's also very important for the DNA repair. This process of ubiquitination is involved in the DNA damage response. In DNA repair, that is basically the DNA damage response. Whenever there is a damage in the DNA, it's need to be repaired. So this process of ubiquitination, it is involved in the repair mechanism. Let me write that this process of the ubiquitination, it involves in the DNA repair mechanisms. Next is the it is also involved in the cell signaling. Now this ubiquitination can regulate various signaling pathways. That basically includes you can say inflammation, that is include immune responses or receptor trafficking. Now this basically includes inflammation. Let me write inflammation, immune responses, etc., as well as receptor trafficking. Now it also involved in the endocytosis. For example, if we simply tag up target protein with the single molecule of the ubiquitin, that is the monoubiquitination. It can basically trigger the internalization of the proteins and the receptors of the cell membrane. That process basically the endocytosis. Similarly, ubiquitination can also modify the histone proteins. So these are the various cellular processes, and this process of the ubiquitination is very crucial for that proteins. Now this basically this ubiquitination, it involves the covalent attachment of the ubiquitin to a target protein. Now let's say this is the target, this substrate is basically the target protein, that protein which we want to degrade, that proteins need to be degraded. So here we will call this target protein as a substrate. Now here this protein, this substrate will be tagged by the ubiquitin. Ubiquitin is a small protein. So this is the ubiquitination tag. After this, this needs to bring in the cytoplasm. In cytoplasm, there is a specific protein, that is the, you can say it's the uh, proteasome, that is the large protein complex itself. That is the large protein complex that is present inside the cytoplasm, and that is basically involved in the degradation of the proteins, that is basically involved in the breakdown of the peptide bonds. Let me write, it is basically involved in the breakdown of the uh, peptide bonds. So these complex proteins that are already present inside the cytoplasm, that will break down the, that will degrade the proteins which is particularly tagged with the ubiquitin. So those proteins which are having the ubiquitination tag, these are being degraded by the proteasome. So this here you can see this is the substrate protein, this is the target protein where the ubiquitin is being added, this protein is being tagged with the ubiquitin. Now this proteasome is you can say kind of a shredding machine which will degrade this particular protein, which will basically shred this particular protein. Let me write, it will shred this particular protein. Now after the protein that was need to be degraded, it has been degraded with the help of the proteasome, and that specific tagged protein is basically going to degrade. Now the question is, what is basically ubiquitin? As I told you earlier, that this ubiquitin is basically is going to covalently attach with the target protein. Now, how does this particular ubiquitin is attached to this protein? Let's say this is the particular structure of the ubiquitin protein. It is having the N terminus, it is having the C terminus. Now it consists of the many lysine residues, especially these are the lysine 63 and the lysine 48. Now if the ubiquitin molecule is get attached to another protein, let's say here we draw another protein, that is the, you can say it's the target protein. Now this protein is need to be tagged with the ubiquitin molecule. Let me change the color. Now this particular protein is need to be attached with the this ubiquitin molecule. Now this ubiquitin molecule will attach to the target protein by attaching its N terminus of the lysine residue. Let me write here. Now this is basically covalently attached to the other protein. Now this carboxyl end of this small protein, that is basically linked to the target protein through the isopeptide bond. What does that mean is, the carboxyl end will attach to the Now this is the C terminus. It is the carboxyl end of that particular protein. This carboxyl end will be attached to the this carboxyl end will be attached to the target protein with its N H group of the lysine, and this particular bond is referred to as the isopeptide bond. Now, this uh, ubiquitin is basically itself is a small protein that is made up of the 76 amino acids, and it is highly conserved in all eukaryotic organisms. It can form the chains on the target proteins that I already told you, that protein that is need to be degraded, that misfolded protein, that damaged protein, which need to be degraded. So this it can form chains. What does that mean is there are several lysine residues that are present on that target proteins. Now this if a single ubiquitin molecule get attached to the any one of the lysine residue of that particular protein. Now this is the monoubiquitination, and if multiple ubiquitin proteins are attached to the target protein, then this is referred to as the polyubiquitination, or you can say it is the multi-ubiquitination. Now what is the monoubiquitination? When there is the single when single you can say when single molecule ubiquitin protein ubiquitin protein gets attached, covalently attached. And what is the polyubiquitination? That is the polyubiquitination when multiple multiple proteins, when multiple ubiquitin proteins gets attached to the target protein, attached to the target protein. So this is the polyubiquitination, and this is the ubiquitination tag that is basically that will determine the fate of the target protein. For example, if we simply attach this single ubiquitin molecule to that particular protein, now that protein will not degrade. If only the chain of the ubiquitin molecule will get attached to the target protein through isopeptide bonds, then this particular protein is basically uh, will be degraded by that particular protein. So this is all about today's lecture. If you like the lecture, then please don't forget to like the video and subscribe my channel. Thank you very much. Allah Hafiz.